Let's get started with cell communication. So, in life, living things need to communicate, even at the cellular level.
In an organism as simple as yeast, there is still cell to cell communication between the two cell sexes, which are known as A cells and alpha cells.
The alpha cells will release a chemical signal known as an alpha factor, which will be received by the A cells, while the A cells will release a chemical signal known as an A factor, which will be received by the alpha cells. This leads the cells to grow towards each other and eventually fuse.
On the surface, this may seem rather simple. However, there are numerous ways, each with many intricate steps, through which cells communicate.
The ways that cells communicate is dependent upon their distance from each other. Cells are either in physical contact, nearby, or in entirely different parts of an organism.
When cells physically contact, they will communicate through gap junctions or cell to cell recognition.
Animal cells use gap junctions, which connect the cell membranes and allows molecules and impulses to pass through a regulated gate. In contrast, plant cells use plasmodesmata, which is a narrow thread of cytoplasm passing through the cell walls of adjacent plant cells.
When cells want to communicate locally, there are two main ways: paracrine signaling and synaptic signaling.
In paracrine signaling, cells release local regulators, which are received by surrounding cells and normally regulate processes like growth or cell division. On the other hand, in synaptic signaling, cells known as neurons, which are nerves that receive nerve signals, propagate nerve signals along their long tails, known as axons, and eventually release those signals in the form of neurotransmitters, which cross tiny synapses to other neurons, which further carries the signal.
When plants and animals need to communicate between cells that are distant apart, they rely on hormone signaling, in which specialized endocrine cells release hormones into bodily fluids, primarily blood, which then flow through the body until they are received by target cells.
However, in order for a signal molecule to communicate with such complex processes, the cell must partake in a three-step process known as reception, transduction, and response.
Cell reception is probably the most intricate step, so listen up. With millions of signaling particles and billions of cells, cells have to have specific reception methods to ensure that they receive the right signals.
In cell reception, there are signal molecules known as ligands, a general term for a small molecule that specifically binds to a larger molecule.
Most of these ligands are water soluble, meaning they cannot go through the cell's membrane. So, they have to be received by transmembrane proteins. The main types of transmembrane proteins are G protein linked receptors, tyrosine kinase receptors, and ion channel receptors.
G protein linked receptors are pretty aptly named as they are receptors that, once activated by a ligand, activate a G protein, which in turn activates another enzyme, eventually leading to a cellular response.
Tyrosine kinase receptors, like the ones shown in the yeast example, are used for growth and reproduction. An inactive receptor system is composed of two receptor molecules. Once they are activated, they join together, which in turn activates the tyrosine kinase region of the receptors, allowing them to phosphorylate other enzymes.
Finally, the ion channel receptor, once activated by a ligand, opens, allowing ions to flow into or out of the cell, which changes the internal concentration and triggers reaction within the cell.
However, some ligands are lipid soluble, meaning that they can diffuse through the cell membrane and are received by intracellular receptors. These activated receptors can directly attach to and control the transcription of genes, skipping the complex transduction pathway.
Yet, receptors on the membrane still have to transmit information from the membrane to the DNA, which is known as signal transduction pathway.
Once the receptor molecule phosphorylates, or activates, a relay molecule, the protein in turn activates a protein kinase. The active protein kinase transfers a phosphate molecule from ATP to another inactive protein kinase, which in turn activates another protein kinase, and so on until the cell signal reaches the final response.
The signal transduction pathway is essential for amplification, regulation, and specificity. Each protein kinases activates multiple protein kinases after it, eventually leading to thousands of activated proteins from one single molecule.
In order to regulate the cycle, protein phosphatases inactivate the protein kinases. The balance of kinases and phosphatases is a way the cell can regulate the strength of the signal received.
The complex set of internal and external proteins needed in order to lead to specific responses allows for the same signal ligand to be around millions of different cells in the human body without triggering deadly adverse reactions.
But not all components in the signal transduction pathway are proteins. Non-protein, water-soluble molecules, known as second messengers, can spread throughout the cell. And the two most widely used second messengers are cyclic AMP and calcium ions.
Cyclic AMP, or CAMP, plays a major role in controlling enzyme catalyzed processes in living cells. Cyclic AMP is generated once an enzyme, adenylyl cyclase, is activated by G protein. This second messenger then affects change within the cell.
Calcium ions are released by secondary messengers diglycerol, DAG, and inositol triphosphate, IP3, which trigger the release of calcium through ion-gated proteins from the endoplasmic reticulum. The calcium, like CAMP, affects change in the cell through changing ion concentration.
Finally, the proteins activated by the signal transduction generates a response.
Oftentimes, this response triggers the activation of enzymes that could, as an example, break down glycogen in liver cells.
Other signals activate transcription factors, controlling the creation of enzymes rather than the enzymes themselves.
And that should be all the concepts you need to know for cell communication.